Our discussion of the ear will deal primarily with the inner ear apparatus. We'll spend quite a bit of time on what's going on in the innermost part of the ear mechanism in there. But to begin with, we'll talk about the outer portion and middle ear portions. First of all, we know what our outer ears like, and it's sometimes called the auricle or the pinna. Auricle is a general name for a flap-like structure, so sometimes it's called auricle pinna, P-I-N-N-A. It's another name given to it, or just external ear. The external ear is probably the best term. There's an opening into what's called the auditory canal or acoustic canal. So here's this opening. Sometimes this opening right here, this is the auditory meatus. Meatus are opening into the auditory canal. And at the end of the auditory canal, it terminates at the eardrum, or what we also know as tympanic membrane, right here. Tympanic membrane or eardrum. So that's all external ear, the opening, the canal, and the ear flap on the outside or external ear lead all the way down to the tympanic membrane. On the other side of the tympanic membrane, we have the middle ear cavity. The middle ear cavity, that middle ear cavity is connected to the pharynx by way of the eustachian tube. There are three middle ear bones that convey a signal across to the vestibular portion of the inner ear, and there's this connection in the same way that there's a tympanic membrane between the outer ear and the middle ear. Between the middle ear and the inner ear is what we call the oval window. Now, sound waves are transmitted through our auditory ossicles, which are a fancy name for ear bones. Here, there are three bones that convey that signal. The first one is called the malleus. The next one is called the incus. Malleus, incus, and then finally the stapes. They are more commonly known as hammer, anvil, and stirrup. Hammer, anvil, and stirrup. So the stirrup the stapes makes contact with that oval window, with that plate right there, to vibrate that plate. Malleus, incus, and stapes. Middle ear bones. Middle ear bones. So vibrations of the tympanic membrane will shake those middle ear bones and transmit that vibration to the oval window against the inner ear apparatus. And that's where we'll go next, just looking at what happens on the inside of the inner ear apparatus. All right, this drawing here represents our inner ear structure. Recall that we had a connection between the middle ear and the inner ear apparatus as an oval window here. Let me move that down just a little bit. Down in here, there we go, that's a little better. So there's an oval window where our stapes would vibrate against this inner structure here. The three portions of our inner ear include these semicircular canals, these loops of tubes out here. These are all semicircular canals. Semicircular canals. This enlargement right in here, near where our oval window attaches here, also includes some inner chambers. We can refer to this as the vestibule. And then this section out here that we know as the cochlea. That snail-shaped structure out there, the cochlear structure. So those are the three parts of our inner ear apparatus. Outside, near the surface of this, there is a kind of fluid that's called a perilymph, and then there's an inner structure, an inner chamber of fluid that's referred, and the fluid in there is referred to as endolymph. So we need to separate out our external chambers and our internal chambers for our next drawing. All right, now I've outlined in green our endolymph chamber, the endolymph portion, and the endolymph is inside each of the semicircular canals. There's a loop of that endolymph fluid, and there's not much space outside of that endolymph fluid for perilymph in these tubules up here. In the vestibular region, there are two separate chambers of endolymph, what's called a utricle and a saccule in the vestibule. Utricle and saccule portions, they contain endolymph. That endolymph continues into the cochlea here as the cochlear duct. So we have semicircular canals, an endolymph portion there. In the vestibule, we have the utricle and the saccule, and in the cochlea, we have this cochlear duct that all contains endolymph. So those are all endolymph chambers. Outside of that is perilymph. Now in the cochlea, that perilymph is divided between two regions. That cochlear duct and the other tissues associated with it divide the cochlear shape here into a vestibular portion, here, this upper part, and then the other portion down here is referred as the tympanic portion. And they go by the names. Let's see if I can do this here. This outer part here, scala vestibuli, and then the other portion over here is scala tympani. Now remember that there was an oval window situated up here, up next to our vestibular portion. In here somewhere, we've got this oval window. Kind of shade that in a little bit. That's where the stapes, that middle ear bone, makes contact with the inner ear structure, and that's where the vibration of sound waves is conveyed to this portion of the ear. So in this fluid of the scala vestibuli out here, we initiate a sound wave, a compression wave, that travels through and eventually back out around in the scala tympani portion to the outside here. So a compression wave travels through, and sound consists of a series of compression waves. Between the compression waves would be a region of rarefaction. So anyway, these compression waves travel their way through here, and at the other end of this pathway, right in here, is another window. This window right here is the round window. And it's thought that it will just allow that free movement of the fluid within the cochlea so that sound waves can travel without being impeded by an enclosed space quite so much. So that's a little window to help. It just gives a little bit to allow that movement. So that's the direction of the sound wave through the cochlea. First down the scala vestibuli, beginning at the oval window, travels through there, then loops around and comes back out the other way through the scala tympani portion. Here in a second, we're going to look at a cross-section of just one part of the cochlea. So we're going to cut through here, just make a slice of that structure there and see what it looks like in cross section to help understand how this vibration wave traveling through this perilymph fluid out here is going to be translated into sound that we hear. Okay, here we are inside the cochlear structure now. We're looking at this whole thing is just one part of that looping coil that makes up the cochlea. At the top we have the scala vestibuli filled with perilymph fluid. Down below we have the scala tympani, also perilymph fluid. And in the middle we have endolymph in the cochlear duct. So here's the cochlear duct between the scala vestibuli, scala tympani here. The vibrations were traveling down that direction, let's say through the scala vestibuli, and then they move back this direction. So a compression wave coming back this direction is going to put force upward on the floor of this structure right here. And this is referred to as the basilar membrane. The basilar membrane, right there. There are cells that lie on top of that, right in here. I have two different colors of cells. Those that are in blue are supportive cells. Those that are in red are hair cells, and they are referred to as hair cells because they have these hair-like stereocilia up here. I'm going to go ahead and label that stereocilia. And these cells are called hair cells. These other cells are supportive cells. So two cell types right in that general region where you get that movement. So here's the basilar membrane pressing up. It presses up on these cells. These cells are pressed up against this flap that extends out here. This flap is called the tectorial membrane. Tectorial membrane right there. So as this is raised up, this is pushed up, and it causes all those little stereocilia to be shifted over, in this case, the tops to the right, as it presses up against that tectorial membrane. So every vibration that comes through here pushes up on that membrane a little bit, and causes these cells to be pushed up this direction, and in the process it causes that tectorial membrane to kind of push against those stereocilia. It's that movement that causes these cells to undergo a depolarization. That depolarization results in neurotransmitter release right down here at the base of each of the hair cells. That neurotransmitter stimulates a nerve fiber. These are nerve fibers here. Nerve fibers that reach out to the base of the hair cells. So that's how vibration waves are translated or transduced is the better term there, changed into neural signals. You get deflection of stereocilia induces depolarization of the cell. The depolarization in the cell results in neurotransmitter release down here at the base. And that's how you hear things. I forgot to mention with respect to this diagram here that we have a fancy name for this portion of the hearing apparatus that is responsible for nerve transmission, the whole neural structure in here. All of this, you kind of make a circle around it here, all that right there. It's referred to as the organ of Corti, C-O-T-C-O-R-T-I, organ C-O-R-T-I. It includes the hair cells, the supportive cells, the tectorial membrane and basilar membrane, that structure right there where we transduce, we change the mechanical vibrations from hearing into a neural signal. That whole thing, that's your organ of Corti inside your cochlea of your inner ear. Now it's worth mentioning that where this basilar membrane is narrower and stiffer, you're going to receive better transmission of the higher frequencies as opposed to lower frequencies. And where the basilar membrane is more broad and more flexible, you get better response and more movement in the lower frequencies. And so different regions of your cochlea will respond differently to the different frequencies. That gives us a full spectrum of auditory receptivity. So let's set up our camera over here. In this portion down here near the round window, this portion over here, here's where you're going to have a stiffer and shorter basilar membrane. This is where you're going to see, where you're going to hear higher frequencies. And as you progress further and further around toward the end, over here, you're going to have a broader and more flexible basilar membrane and be able to respond to lower frequencies over here. So your high pitch to low pitch is a gradual change from one end to the other in the cochlear structure. So that's how you interpret different frequencies of pitch.